Right, this is the Chapter 7 video control of microbial growth. In this chapter, we discuss methods to kill or reduce the growth of microorganisms. Ignaz Semmelweis and Joseph Lister were two individuals that were instrumental in setting the stage for microbial control methods. This was in the mid 1800s. Before these methods, such as hand washing and aseptic surgical techniques, 10% of deaths were due to nosocomial infections. Mothers delivering babies had a death rate as high as 25%. And during the American Civil War, a field surgeon may have cleaned his scalpel off by wiping it on his boot between patients. So some terminology, sterilization would be removing all microbial life. This would include what are called prions, which are infectious proteins. These are known to be highly resistant to all forms of sterilization, including autoclaving. The definition of sterilization implies the absence of prions as well as microbes and viruses and fungi. Disinfection would be removing pathogens from surfaces or substances. And then antisepsis would be removing pathogens from living tissue. So first we'll look at some physical methods of controlling microbial growth. The first one being moist heat sterilization. What happens with moist heat, what it does is it denatures proteins. So proteins, as we talked about, are important for all cell functions, enzymatic reactions, biochemical reactions. We can use heat to preserve canned foods, which is the most common method of food preservation. The autoclave is used for medical instruments that aren't heat sensitive. We use this for our lab media, our glassware, and things like that in the lab. Autoclaving provides 15 pounds of pressure per square inch at 121 degrees Celsius. And that kills all organisms, including endospores, but not prions. There's dry heat sterilization, so basically using an oven type sterilization method, which kills by oxidation. So there's different methods. There's dry heat ovens, there's flaming, incineration, and hot air sterilization. The equivalent treatments from hot air versus the autoclave, you need to go for 2 hours at 170 degrees Celsius versus about 15 minutes at 121 degrees Celsius for the autoclave. Pasteurization is another heat method, which is going to reduce the spoilage organisms and pathogens in certain foods. So this is a high temperature for a short amount of time. 72 degrees Celsius for 15 seconds would be useful for things like your milk products. There's also ultra high temperature, which is 141 degrees Celsius for less than a second. So your organic milks are commonly pasteurized with this method, including coffee creamers. Low temperature will simply inhibit or slow microbial growth. So refrigeration or deep freezing, just providing suboptimal temperatures for microbial growth. And this makes sense if you take a hamburger out of the freezer, for example, and leave it sit in your refrigerator too long, that hamburger will still spoil due to microbes present within that hamburger meat. Filtration is another physical method where we're going to physically remove microbes from a media. So either from the air with HEPA filters, membrane filtration from liquids will remove microbes as small as 0.22 micrometers. So membrane filtration, HEPA filtration is good for most microbes, but not viruses. Viruses are 5 to 300 nanometers. So this is 300 nanometers is about 3.3 micrometers. Influenza viruses are about 125 to 150 nanometers in diameter. So membrane filtration and HEPA filtration would not necessarily remove viruses from the air or from a membrane filter. We've done this in the lab where you've poured the liquid media through the membrane filter, so all those microbes were trapped, and then this media that flows through the liquid is sterile. Radiation is another physical method where we're going to damage DNA. So there's three types of radiation: ionizing radiation, which are X-rays and gamma rays, damaging DNA. So we can use this for things like spices, meats, and vegetables. Some of the meat that you buy at the grocery store would have gone through a gamma radiation treatment. Disposable dental and medical supplies such as plastic syringes and surgical gloves are also sterilized using radiation. The post office uses uh, ionizing radiation in certain classes of mail. Non-ionizing radiation is ultraviolet rays, which damages DNA but does not penetrate surfaces, so it's only good for surface applications. And then microwaves would actually kill by heat and is not really antimicrobial. Here's the image with strawberries. So these would have been picked at the same time using irradiation. The strawberries stay fresher for a longer period of time. Where non-irradiated, those would succumb to fungal infection and spoil very quickly. Some chemical methods of microbial control would these chemicals alter the permeability of the cell membranes and damage the damage from that cell membrane results in the cell contents leaking from the cell. Also, they can damage proteins, so they can denature proteins or damage nucleic acids as well. This is a list of the most resistant microbes to the least resistant. And pay attention to basically the bacteria here. We'll discuss prions in Chapter 12. But endospores, as we talked about, are highly resistant. Your mycobacteriums with the mycolic acid, gram-negatives with outer membrane, and then your gram-positives with no outer membrane but just the cell wall would be the least resistant. So the microbial characteristics affect the control method. Gram-negatives, like we said, are more resistant than gram-positives. And Pseudomonas, for one example, is very resistant to most disinfectants. And that outer membrane is what's going to provide that protection because those porin proteins in the outer membrane only allow certain molecules into the cell. Your Mycobacterium tuberculosis, other mycobacteriums, those have the mycolic acid, which is that waxy lipid component of the cell wall resisting the effects of chemicals. Here's an example with Staphylococcus, E. coli, Pseudomonas. So E. coli, Pseudomonas are both gram-negatives. You can see Pseudomonas is resistant to 3 of the 4 chemicals here, where E. coli is resistant only to 1, where Staph aureus, those chemicals would have an effect on killing Staph aureus. Like I mentioned, we're not really going to be too concerned about prions right now, but prions, these are the most resistant form of microbe known. These are infectious proteins. They're autoclave resistant. They're protease resistant. Protease is a enzyme that breaks down proteins, and these are known to be resistant to those proteases. So in order to destroy them, they have to be autoclaved in a sodium hydroxide solution or incinerate them. So basically with chemicals, I mentioned before, you don't have to memorize all of the chemicals for this unit, but there's no single disinfectant appropriate for all circumstances. The method used to control the microbes are good at preventing disease. However, the disinfection procedures are usually never going to 100% remove all of the microbes from a surface. You hear this on commercials. Lysol kills 99.9% of all bacteria. There's always going to be some microbes that will be able to resist the effect of that chemical. Now, if you're familiar with antimicrobial soaps, antimicrobials that you use in the household, basically there is no evidence that those products aid in human or animal health. And actually, those products just promote the development of resistant microbes. So the use of antimicrobial soaps, for example, should be limited to food handlers, those caring for newborns, and for high-risk patients, such as those who are immunocompromised. The CDC recommends using regular hand soap, washing with warm water for at least 10 to 15 seconds to physically remove those microbes from the surface. One of the products that is added to um, certain household items would be this chemical called triclosan. Triclosan has been banned from the use in hand soaps, but there are other antimicrobials that are used in hand soaps. But like I mentioned, hand soap, the hand washing procedures is going to physically remove the microbes. Um, Chemicals like triclosan actually have a 2 minute time where they need to actually be in contact with the surface of your hands in order to kill microbes. So, for the most part, none of us wash our hands for 2 minutes. So, it's completely useless, basically. However, triclosan has been added to other things such as toothpaste, deodorants, um, which again, there is no evidence that this aids in your health. Um, Triclosan has also been incorporated in cutting boards, knife handles, it's in dog dishes and things like that. And widespread use has resulted in reports of resistance to triclosan. All right, this will conclude the Chapter 7 video, and there will be the quiz available on e-learning for you to complete as well.